So our next speaker is from Morgridge and Juan Kakaido's lab, John Peters. He's going to synthesize several things that have always been touched upon a little bit. So it's always interesting when earlier things come together. He's going to talk about AI, high throughput computing, and even a little bit of Snakemake. So sadly, Justin is not here. He would be very happy.
He's here.
Oh, there he is.
Yeah, he's here.
Okay, thank you, John.
Okay, okay, I can hear myself. We're good. So thanks for the warm welcome. Like you said, I'm in the Caicedo lab, and I'm John, and we're going to talk about how we utilize CHTC to more easily churn through a couple terabytes of data in a Snakemake workflow. So we're an imaging lab, and so what that means is we have tons of pictures, and the sort of end goal for all of this is we want to identify the left pictures from the right pictures, but to do that, these pictures need to be in vector space. And to get these pictures into vector space, we use deep learning. And so we're going to be talking about how we used a ton of CHTC jobs to get a couple million images into that vector space. So to give a little bit broader context, these image-based workflows, they do start with this feature extraction step where we're putting things into vector space, and then we prepare the data so that we can do like statistical tests on it so that we can identify the differences between those pictures. But for this talk, we're just going to talk about step number one. We're just doing a ton of vector space work so that we can do the statistics later. So for this talk, we have 5 different studies that we were processing so that we can get these images in vector space so we can do our little stats. These 5 studies are stored in Data Vault. So in Morgridge, we're 1 floor up from CHTC, and in the building we have shared storage that works sort of like the research drive that the previous presenter was talking about. And it lets all of the jobs like push information and pull information from the shared space. It's essentially staging, but it's what we use. So our data organization here, relevant to how we can parallelize this, is we have our images in a directory in this mounted drive. And then we have the different 5 little studies have their own directory. And then in there, we have zip files of all of the images. And each of these studies have a variety of different ones. At the very end, I'll tell you the actual numbers of how many of these we went through. So the format of these, so I said we had zip files, and these zip files do contain images. We have them sort of containerized so that every single individual image is multiple discrete image files in these zip files. And they're all in the same zip file. So we can go from the full image, we can take the individual different channels, which in fluorescent microscopy, these individual channels often are relevant biology of interest. So for this study, this picture is IDR88. It was in the bottom right in the last slide. And the first channel of it is just a stain over the cell body. And then the 2 other ones are different protein markers that they use to identify the differences in the picture mostly. And so these are in individual PNG files in this zip file with no compression. And then we also have like metadata that's associated with this so that we can really easily collect those 3 images from the zip file, package them together, and do like multi-channel inference and stuff. So the big overall pipeline that we'll like dive deeper into is this sort of 4-step pipeline where we got to get the images from Data Vault onto the actual compute nodes. Once we do that, we have to use a segmentation model to get masks that outline the different cells that are on the image. And after we do that, we'll take the images and the masks and we'll get individual cutouts for all these cells. And then these individual cutouts are sort of like the batch that goes to our deep learning models that we'll be talking about. And so the images really isn't like the scale of what we're predicting at. It's like the total number of cells are actually what's going through these models. So it really increases the number of steps that we have to do to collect all of these features for all of these images. And so then the output of that is just a little matrix that describes all of the cells on the individual image. So you could, conceptually, you can be like, okay, we have these individual zip files. We have however many of them. In our case, it's a few thousand, but we could send that whole pipeline of those 4 steps as one little CHTC job. We can wrap it up in a queue job, and then we can just spam the submit server with many, many jobs, and that would work. If you had like 1 model that you wanted to test, that's fine. Those steps don't need to be cached. It's perfectly fine, but for what we were doing, this is kind of slow. And so we wanted to speed this up. And we can speed this up by caching the first step. That segmentation step is actually the same for if we want to test one model, two models, three models, five, however many runs we want to do of this pipeline, this segmentation is a constant. And so we can go through and we can get these segmentations, save the masks, and then we can just load them later. And that whole step of like running a different deep learning model to get masks of our images, we don't have to do. And that saves us tons of time because the segmentation is like twice as long to do than just the computation of the model features. So we want to get that out of the way because it's constant. And so now though, we can't really do as easily those queue jobs unless we want to like manually be the ones running the pipeline. And so we're computational people and we're using Condor. So we can have it do like a lot of the heavy lifting for us. So we don't have to be the ones to be like, okay, run this segmentation job and then go over here, run this model and this model. We can just have it do it all for us in the order that it designs like it wants to. So we use Snakemake for that. So Snakemake is a workflow manager. It's similar to Dagvan, but it lets us do like fun things like we can run our pipelines locally. And so when we're testing and developing, we can run these Snakemake jobs the same way they would get executed on Condor, but on our local servers. And so we can test and like verify and make sure all of the outputs are saved to the right directory and the right structure, and they're in the right format and they're good. And so it makes it really easy to use the good thing of CHTC is we can just like spam a ton of things, but it lets us do a lot of the hard, annoying work of like debugging locally, and it saves us a bit of time. So conceptually, the Snakemake job will have two parts. It'll have the part that gets cached and then the actual feature extraction part. For this, we can zoom in and see what the actual like job order of things that the Snakemake task is running. And it's kind of simple. It's also 4 steps, but we download the zip. So one of those zips that you saw in the study directories earlier, we're just going to grab one of them. We're going to unzip it. A lot of our data loaders don't play nicely with zips just because of concurrent issues with how like PyTorch forks. And so we like to unzip things. And then relevant to that separate channel storage earlier, we can actually save a bit of IO time here by just loading 1 or 2 different channels for the segmentation. A lot of the time we just care about like where the cells are, not the actual like specific shapes around the cells. And so we can speed up our life a little bit by just like grabbing 1 channel. And so we can have like a third of the IO bandwidth that we need to use there. And so that's happening on the compute node. We take that individual channel, we give it to a segmentation deep learning model, and then it gives us an image of masks out. And so these are PNG images, like 16-bit, and then they just have like the brightness of 1 is the first mask and 2 is the second mask and so on. And so you can sort of see from the top to the bottom the cascading of more brightness because the later masks end up in the bottom of these images. And so those masks in the same format with the same names as the original like zip file, we can save in Data Vault as well. And so this sort of naming scheme of just being another zip file with like slightly the same name, just with like segment tacked onto the end of it, lets us super easily like get the masks for the specific image, and it's very reproducible and very understandable for how we can like quality check these, because we do have to do that. So that lets us have these masks now in Data Vault. And so we can take with our feature extraction step, and in parallel, we can extract those features. And so we can do them across many different models. And the joy of Snakemake here is we can have Snakemake run the masks for one model, or if we don't have masks and we want to start with one model, it'll just go and grab and do the compute for those masks. But it'll just submit all the jobs. So what I tend to doing is for each model, it's one Snakemake job on the submit node. And so I'll go through, run the segmentations with one of the models, and then when that's done, I'll just like spam the other 4. And I'll have usually like the ones I was working with were a couple hundred jobs just waiting to get queued, and then they'll go through like, I don't know, like 40 at a time on GPUs, and they'll go through. So if we zoom into this, it's also a very simple thing. So we compose these more complex, like larger job into these very 2 simple jobs. And it takes the information, so the images and the masks that we saved previously. It unzips those both. So we have the masks and the images. It chops out the individual cells from those masks. gives them to the deep learning model, and then we pipe that right back out to Data Vault. And so this is a very handy way for us to get all of those features. And I did say that this was supposed to be large scale, so we can talk about scale. Of the zip files, we call them plates, usually internally. We had a bunch of them. For this study here, 88, there's like 1,200 of them. That's 1,200 zip files that we sent through this pipeline. For IDR9, that's 1,000 of them. And then these other ones like 17, 20, and 7 they're like less than 100, so it's not too big. But in total, we were able to have like tons of data go through. And so that's like the x-axis. And then the y-axis here is the total number of gigabytes. And so we can see when we look at studies like IDR 88, we're working at 1.5 terabytes. And so this is 1.5 terabytes that goes through that pipeline. With 9, that's another half terabyte. With 17, that's another half terabyte. And with these, it's like the total number of gigabytes here is like 30. But in total, we had around 3 terabytes of data just turned through all of this pipeline, which was quite nice. And it took about a week. And we have a couple servers there with priority access. So for the GPU scheduling, we usually get 8 GPUs per server. And so we have about 4 servers there. This note right here is for one thing that's kind of interesting with this type of pipeline is that you note that 9 and 17 are the same number of gigabytes, but the number of zip files is vastly different. And that's sort of because this IDR17 study over here, it has much larger images, and they're all packed together in larger zip files. And what this sort of means is that the resource allocation for these Condor jobs is higher. And one thing that it happened to do was if our servers are 8 GPUs, 96 CPUs, and like half a terabyte of memory, these jobs started using more memory than is like splittable by 8. And so then sometimes when these jobs would go on a server with 512 gigabytes of RAM, I can only fit like 7 jobs on there. And so if you're working with 7 jobs on there versus if you can pack them full with smaller jobs and you can get 8 on all of them, you start to lose some of that efficiency because the servers can't allocate another job on there just because there's no more resources, even though it has the GPU. So that was one sort of important consideration that we had when we were working with these like dynamic types of studies, is that we had to be careful on how these jobs went through so that we can maximize the throughput of the servers that we were allocated for. Additionally, because these were priority servers, we have other people in the lab that were also like wanting to use Condor jobs on them. And we found like with the Snakemake executable, if I set it to submit 150 jobs at once, the next person would have to wait for all 150 of those jobs to complete. And so they might be waiting for like 2 days to get a slot on here. And that wasn't really ideal. So we found that submitting lower batches of jobs in like the 30 job range so that we can fully saturate the servers and then still have people, when those jobs die out, like have their job go in like a couple hours instead of a couple days was really good.
Did you consider using job priority to maybe address some of it? That when you submit a batch of jobs and you want somebody else to come in and not follow a FIFO order, that they use a higher priority, something along these lines. Did any of this ever come up in your discussion? Are you aware that different jobs can have different priorities?
Yeah, for us at least, on our servers, we were just having all of our jobs from everyone in the lab have the same priority, and it's just FIFO. But since it took a week to process the 4 terabytes that we were working with here, it wasn't too bad to just have one server dedicated to training and the 3 others were just computing features.
Okay.
But it is a solution.
Or at least something to explore, and if we need to do better or different on this is so yes, FIFO can be annoying.
Yeah. But because we only have like 40-ish GPUs in that pool of priority servers, if we set the amount of jobs that Condor is willing to queue at that number, they'll drop off in the queue quick enough that a job that's just added it doesn't have to wait too long. Okay, so what we sort of learned with this is with CHTC especially, we think of a lot of these resources as just like isolated chunks. But when you are trying to put as many jobs onto a machine at once, it's important to consider the like total impact of the resources that the machines have, just because you might not be able to fully saturate them, and then you're waiting longer for your results. The Snakemake addition that we had help with was very good for locally testing these pipeline runs, and then also allowing them to just spread out and go crazy on CHTC. And then the sort of most important thing that we did during this was the caching of the segmentations, just because the output of this is we're in the 6 terabyte range on disk, not including the images, just the segmentations and features. And if we're generating like 3 terabytes-ish of data every time we have to compute features for our next model, it sort of goes very slow, and it would have probably taken us like 3 weeks instead of 1 week. Okay, well, that was what I was discussing. This is the lab I'm in, my PI here, and then special thanks to Justin in the back and Ian as well, because they helped us a lot. We met like once a week, every week, so that we can hash out all the issues with Snakemake and like Data Vault and all of those things, and it worked very well. And then thanks for the NSF for giving us the money for projects like this, and I'm funded on a training grant. Thank you.
Questions for John?
Hey, John. Thanks for the presentation. I'm almost maybe a little ashamed to ask this question that I don't already know the answer, but I think in the process of getting your science to run in CHTC, we've run into a number of different types of road bumps and roadblocks. Can you maybe say a little bit about kind of which of those that we've encountered you would identify as ongoing, or the biggest things that if we were able to solve them for you would kind of unlock that next level of what you're able to do with your science?
Honestly, the biggest thing was when we got Data Vault access. That was the best thing. Like the Snakemake, getting it working was fantastic, huh?
What was the first thing?
The first thing was Data Vault there were some issues with mounting on these.
Shared file system.
Yep, the shared file.
The shared file system are evil.
Yeah.
They are declared and recognized as pure evil.
But we slayed the evil, and in our hero's journey, we did prevail, and getting the shared file system so that Snakemake could see that the segmentation jobs were actually cached and wouldn't rerun them was fantastic. And having Data Vault be our shared file system was nice, just because we have quote unquote unlimited storage there. And we were hitting like 2 terabyte, 5 terabyte walls with staging, and so it was nice just to have no limits. But that was the biggest thing. I think that was the.
But the downside of depending on mounted file system is that you cannot go anywhere else. It limits if somebody calls you and says, I have here another 10 GPUs, you cannot do anything there, because then you say, hey, we have to mount, you have to do. The question is there a way to run this without the mounting?
Yeah, if we were willing to, I know on campus we have like our like pseudo AWS storage type stuff. If we were to mount it in a remote storage, that it could quickly.
Mounting is not
no, not like
is not scalable. You know, if there are 20 GPUs in San Diego.
Yeah, but if we hold things if we move our images to like a local AWS type bucket and we're pushing the output to there as well, we can have Snakemake query that file system, and that happens to the network. So that can just be wherever. And so
Yeah,
it doesn't have to be like directly mounted. Mounted is just convenient for the work we do in the file.
It's convenient, but it's a trap.
One of the issues here is that I have not yet gotten Snakemake to work with Pelican, but that is something I'm thinking about and have maybe some ideas for. So.
Thinking is a useful exercise. But, so when I hear mounting, I say this is not throughput computing.
Yeah.
They should go to the Mountaineer computing.
Thanks, John.
Yeah.
Thank you, and let's have a round of applause for our speaker.